Okay, what I'm going to do before I start some cellular respiration, I am showing you the Campbell Biology 7th edition correlation paper that you can get from Biology Junction. You go to Biology Junction, you click on AP Bio, and at the top of the page you will see this link. What this does, if you will notice, it says here's what to cover and here is what to omit. Now, this is just a quick guide for you guys as far as being able to get through your book more effectively. Let me tell you, especially you sophomores who have not had biology already, the AP people assume a lot, and they assume that you've had a lot more biology than some of you have actually had. So that by omitting some of this, it may or may not get you there, but I will tell you it will make your life a little easier as far as knowing what you need to have for the AP exam. I would suggest going and getting some of those little tabs so you can tab your book out. Okay, what I'm going to do for you right now is going to be a really quick cellular respiration video. And this is basically just to back up what you've already had. And I'm going to draw this for you rather than have a sheet for you already ready. Alright, so this is cellular respiration. Alright, and I hope we can see this okay. I've got it up on the book trying to get a little better. You should see the setup I've got here trying to do this. Alright, so what the deal is that we have the bloodstream. And in the bloodstream, let's pretend that you have eaten. And so now you've got glucose running around inside the bloodstream. Okay, so we're going through the bloodstream, and glucose, of course, needs insulin to get into your cells. That's the thing that's going to help it if you've gotten cell communication already. Okay, so let's call this a cell membrane right here. And those of you that have had me before and we've already done this will recognize my drawing. But there's a cell membrane right there. So insulin is going to help glucose, which is a 6-carbon molecule, go right on in. Glucose can get in anyway, and glucose can get back out. There are channels for this. But for the most part, what's going to happen is as soon as glucose gets in here, it is hooked up with a phosphate group that's going to make it have to stay inside the cell. Alright, there's more to it than that, but for right now, let's just go with the fact that glucose is in there. It goes through a series of steps that are going to keep adding on phosphate groups, etc., etc. But the biggest idea is the fact that glucose is going to have to be broken down into 2 3-carbon molecules. That's the process we're starting. We're going to break it down into 2 3-carbon molecules. The thing that does that is going to be ATP. ATP, it's kind of like you have to spend money to make money. ATP has to come in here, and in the process, it's going to break down this carbon by getting rid of a phosphate group. So you notice that ATP moved in, and ADP is coming out. And when that occurs, you're releasing energy, which helps do that. So you have ATP that you've expended, all right, and you're not making anything, and then you're going to do this again. And as the ATP comes in, then you're always going to release back into the cell ADP. So we have spent 2 ATP. All right, but this 3-carbon right here is not usable. Okay, so we're going to have to do a few more things to that 3-carbon. And at that point, we have an intermediate type of thing that comes in. It's called NAD, or NAD+. And what NAD does is comes into this long chain carbon molecule, not long chain, but whatever, that's going to have some hydrogens on there, and it is going to pull away a hydrogen and hook it on to NAD. So that's where you get the NAD going in, but it comes out of this reaction as NADH. When that occurs, then ADP can go back in and grab those phosphates back away, and it's going to make ATP. Alright, this is going to happen twice on each side of the equation. In other words, you've had these 2 3-carbon molecules, so we're going to get some more NADH out, and we're also going to get out ATP. Now, anytime I write down that we're getting ATP out, ADP had to be there to supply the molecule to get it. Oops, sorry. Okay, so you will notice at the end of all this, I never mentioned the word carbon. So we still have a 3-carbon molecule, but I have earned plus 4 ATP. This is a minus because we used it. So we have a minus 2 ATP and we have a plus 4 ATP. If we had a scorecard, we would now say we were plus 2 ATP to the good. Here's the deal. This whole process right here is called glycolysis. Glycolysis is also known as anaerobic respiration, because at this point, if everything stops right here, if there's no oxygen in this little critter, then this is good enough. We're going to stop the process at this point. So glycolysis is anaerobic respiration, and it's good enough for little things, for example, like yeast. They don't need a whole lot of energy. They're tiny little single-celled organisms. And so by doing this, they're able to get enough ATP. Now what yeast or bacteria will do is to convert this 3-carbon, and what this is called is pyruvic acid. They're going to convert this into, we'll make sure we know that this is yeast down here. We're going to convert this into, if you're yeast, alcohol. Okay? That's why when you have yeast breads and stuff, it smells faintly like alcohol. It also will convert, if you're a little bacteria, you convert this into, say, lactic acid, and that way we can get some sour cream or something out of you. But what happens if you're a big critter like us? Okay? Well, what happens at this point is, if there is big, I'll put big organisms. All right? What happens is that there's no oxygen. We're going to convert this into lactic acid also. And this is the lactic acid that you hear about when you're talking about muscle cramps. Those of you that are runners and swimmers know this part. The deal with this is, if you'll look on this side, I'm a little yeast. I'm going to make alcohol. By the way, that's ethyl alcohol. All right? If I'm a little yeast, I'm going to make alcohol. Once I do that, it's alcohol. If I'm a little bacteria, and I'm going to turn this into lactic acid, once I do that, it's bacteria, that's lactic acid. But in larger organisms, evolutionarily speaking, this is a little more efficient. This is not good for us. This is muscle cramps. It's really going to hurt. But what the deal is watch this arrow. It can be turned back into pyruvic acid so it can continue on down the pathway. All right, so let's go back to the top for a second. All right, we've got glucose as a 6-carbon molecule that comes into the cell. The cell is going to spend energy in order to break this down into 2 3-carbon molecules. At this point, whatever cell it is going to be able to make some energy and some NADH. These are things that are carrying energy in the form of a bond right there. All right. Anyway, we're going to make some energy so that we get an actual little surplus of energy. Not enough for a big organism like us, but certainly big enough for yeast or bacteria. Yeast and bacteria are then going to turn around and take this 3-carbon molecule and turn it into either ethyl alcohol or lactic acid. And by the way, one of the byproducts of this process is going to be CO2, which puts bubbles in beer and bubbles in bread. All right. But the deal is, in the big organisms, is that the lactic acid once made causes us problems. You rub your muscle, makes it red because you're bringing blood to it. Blood brings oxygen. We can convert this lactic acid back into the 3-carbon molecule to continue on down the pathway. And that way, that's more efficient. And we don't live with muscle cramps for the rest of our lives. That's glycolysis. We're going to do a part 3 because this is long enough.